All right, so I decided to spotlight a paper published in 2014, authored by Dr. Austriaco and his lab team at the time. It focuses on the functioning of this antifungal drug called caspofungin in two yeast cell species, and then how that relates to PCD, or programmed cell death. I've actually organized this slideshow so that it follows the structure of the paper pretty closely, so in case anyone wants to read it at some point, I hope this helps make things a little clearer. I also recommend reading the methods and materials section before the results and discussion in the actual paper. That makes things a lot clearer. I'll try to go through this as fast as I can. Some bits can get a bit technical, but I'll try my best to simplify whenever and whatever I can. Okay, so the paper begins with an introduction that basically explains how past research has led to questions that it tackles. It talks about parallels between PCD and yeast cells and mammalian cell apoptosis. So yeast cell research can actually be super beneficial to pharmacology. It then introduces caspofungin, which is an antifungal drug that works by blocking the synthesis of beta-1,3-D glucan, which is a sugar in the cell walls of fungi. So then the paper makes the claim that yeast cells treated with caspofungin will undergo PCD, and that several factors could be used to measure that. Some of the ones they list are the formation of ROS, or reactive oxygen species, lowered mitochondrial membrane potential, and mitochondrial fragmentation. But I think the more important claim is that yeast cells require this so-called AIF1 gene for the PCD to actually proceed. So I'm not going to focus too much on the methods and materials since there's some pretty obscure lab techniques and equipment mentioned. But the gist is that both yeast species had mutant and wild type strains that were placed in control and drug containing mediums. They were incubated and then tested in a number of ways to measure evidence of PCD. Also, feel free to pause the video at any point in case you're interested in the more technical jargon. Okay, so as expected, the lab tests showed evidence of caspofungin's PCD-inducing abilities, but then the paper brings up the idea that PCD has two different pathways, one being caspase dependent, in other words, requiring this YCA1 gene in the yeast Saccharomyces cerevisiae, or the MCA1 gene in Candida albicans, and the other being caspase independent, which has nothing to do with those genes. So I guess that goes on to explain that there was essentially no difference between mutant and wild type strains of yeast cells in a drug containing medium, and that because the AIF1 gene was known to be associated with caspase independent PCD, mutant strains without that gene were also included in experiments that the team did later. And then those experiments actually demonstrated that strains lacking that AIF1 gene were more resistant to caspofungin. And I guess this paper is the first to make a connection between caspase-independent PCD in these yeast cells and that particular gene. Okay. So I know these are pretty hard to see, but I wanted to include the paper's figures that had pictures actually showing the results of those lab tests. Yeah, and that's pretty much it. Again, I highly recommend reading this paper. I think it's surprisingly digestible. It's not long, and it's definitely made me more interested in microbiology research and how it can potentially lead to the development of new drugs. So yeah, that's about it.